So I'm really excited to come and talk about my favorite organ, the brain. And so I'm glad you guys all made it out this morning. At the World Psychiatry meeting last year, I spoke a lot about heroin. Today I'm going to focus on another drug, but really emphasizing the vulnerability of the brain for addiction disorders. And I start off by just emphasizing a couple of things that I think in society we ignore addictions. And we ignore addictions in part because it's not the prettiest disorder in the sense of you look at people who are destitute, who we think that they have caused this problem because they chose to take the drugs. It's a problem about morality and that they have weak morals. However, drug addiction affects a large part of our society. When you look at the numbers in terms of the amount of money that drug addiction costs, it costs more than double what cancer does in the US. Yet still very little money goes into looking at addiction research. And the problem is also that a lot of this is related to the incarceration, the judicial system, that there's a lot of addiction associated with the prison system. So we lock up a lot of people, but we don't really try and find out whether the cures and the treatments that we can help with these disorders. So this is numbers just looking at the drugs that are abused in the US. And I'm sure most of you are not probably surprised. Well, actually, no. I think, are you guys surprised that marijuana is the drug most abused? Meaning that it's clinical diagnosis for abuse. Because many people don't think you can become dependent on marijuana. But it is the drug. You're surprised, yeah. Because many people think you can't become dependent on marijuana. But clinically, it is the drug that more people in the US are clinically dependent on. And obviously, you all know that pain relievers, a lot of opiate pain relievers have unfortunately grown in our society in recent years, which is another huge, huge problem. So it's funny, when I decided to study addiction disorders, a lot of my friends were surprised because I will tell you, I'm a weird person. They said that I have never tried marijuana, and they were like, but you're born in Jamaica, and went to college, and in college all these kids are smoking marijuana. And I remembered when I went to college was when I realized certain things about addiction. My family were very strong in terms of, don't go down that route. And a lot of my friends experimented, and some of them didn't go on to achieve their potential. But I got to college, and a lot of kids were smoking marijuana and doing all the same things. So I realized there are a lot more things to just that. And we have to come back to the environmental issues. The things that are different are the fact, like I said, many people will try marijuana or other drugs. I'm not even talking only about marijuana. Many drugs of abuse, but it's only a certain percentage that have a problem, that have these mental illnesses later. And so the question is, what is different about these people? What do they experience in their lives that make them much more vulnerable? And so this is like the take-home message. The take-home message is that there are many factors that contribute to individual vulnerability. You have aspects of the environment that they are exposed to from prenatal times through adolescence, through adulthood, the genetics that bears on that vulnerability, and to their behavioral traits. And we know that certain people are more sensitive to reward, certain people are more sensitive to inhibitory control, anxiety, and we know that those things contribute. So also psychiatric comorbidity, for example, with depression, and all of that contributes to risk. However, as a neuroscientist, I want to know what is it in the brain that contributes to addictive disorders. So it's not a complete, absolute pathway in the brain, but there is a circuitry in the brain called the mesocortical limbic system. And most addiction researchers study, in large part, these areas. In fact, many people, not just addiction, but mental health neuroscientists will study it. And we study it because these brain regions are important for reward, for cognition, for emotional regulation. And so some words that you'll hear, for example, it's called the striatum. And the striatum modulates, is really important for the reward we feel, our goal-directed behaviors, motivation, and even the habits that we have are all encoded in the striatum. And it integrates information from many different brain regions, including the cortex. And a part of the prefrontal cortex is called, important is obviously the cortex for cognition and for inhibitory control, for example. I point on another brain region, I'm not going to talk as much about it. For example, the amygdala, important for emotional regulation. So this mesocortical limbic system, and in large part the striatum, has been of large focus for addiction. Now, a transmitter even that's highly studied in addiction disorders is dopamine. Who has heard of dopamine? Yay, okay, I can leave now. No, I'm teasing. So dopamine is the quote unquote the reward transmitter, and that's how a lot of people, including me when I was in school, we thought that that was what dopamine does. We now know because of computational biologists. In the beginning, I really did not like certain aspects of math, and I was like, why do I have to study this part of math? And then it became evidence, really cool, that computation can help you understand biology in a way that we really didn't just looking at, say, dopamine and the striatum, and I'm going to come back to that. But dopamine is important for attention, the prefrontal cortex, for, as I said, reward, reinforcement, and those cells that make dopamine, they're in the midbrain of the, and it's in this area called the ventral tegmental area. That's part of this, that originates this mesocortical limbic system, and it projects into the striatum. It projects into the cortex, into the amygdala, and so on, and that's what regulates this wide spectrum of behavior. For a long part, as I said, addiction, dopamine hypothesis was very prominent. And it was prominent in the context that when you took a drug, people hypothesized dopamine went up, and then that was associated with euphoria, the rush that you felt with drugs. And when the drug was gone, and especially if you were a repeated user of the drug, over time it would take away, decrease your dopamine levels, and that made you felt bad, this dysphoric effect. And so people took drugs to increase the reward. But one of the things that people started, as I said, is certain mathematicians started coming into neuroscience in a way and actually studied the patterns and the frequency of the dopamine cells. It became apparent that it's not dopamine that is rewarding per se, it's the prediction. And our brains are computers. It's about computation. Everything we do, from what you're thinking now and what you're going to do later, it's about based on information you had in the past, what then changes or modulates the behavior you'll have in the future. And dopamine is about prediction error. So for example, your dopamine cells fire and dopamine is released when actually you get an unexpected reward. So you thought that you got a B on that exam, and you get it back and you get an A, your dopamine cells fire a lot. That unexpected reward is that's what really triggers your dopamine cells. The opposite, if you expected that you got an A, you got a B, those dopamine cells decrease their firing. So what drugs do when you take them and you increase dopamine, for example, they make the brain think it has gotten an unexpected reward. So remember, dopamine is about prediction errors, and as I said, the field, we're learning more and more about these dopamine circuits, and it's been quite interesting. Another transmitter system that's really important for addiction are the opioids, and the opioid peptides, they're called. And the opioid peptides, many people know endorphins, and so I'm going to keep it simple in that context. There's a whole family of opioid peptides. Like I said, everybody knows endorphins? I hear it. Okay. So many people know of endorphins because people, like when you exercise or something, people are like, oh, you get this endorphin rush. That's never happened to me. I'm still waiting for that endorphin rush. Oh, it's funny. That picture, the colors are gone on that one. But the endorphin, the opioid peptides are expressed throughout your brain. And one of them, enkephalin, is, you can say, your natural heroin. Heroin actually mimics the enkephalins in your brain and endorphins. And they're actually about hedonia, about joy, that kind of state. So today I'm going to focus on primarily marijuana, and how heroin comes into that context. I'm going to focus on it for this reason. I figured since this was going to be an audience of students, and of high school students and graduate students as well, the numbers about marijuana are getting a little disturbing. And even though many states are starting to legalize without understanding the impact of marijuana, I think long term, but for the first time in the history of when we're trying to measure, when we have taken surveys about drug use in the US, more teens today smoke marijuana than they do cigarettes. And they smoke more marijuana than cigarettes because we've done a great job of informing about the health impact of cigarettes and that it's bad. So many teens think that cigarettes are bad, but cannabis is fine and great. So that even like in the US, you have nearly over 30% of 12th graders smoking marijuana. So the fact that they think that marijuana is not of high risk is a huge problem. However, as I said, you said you didn't know you could become dependent on marijuana. Also, marijuana is the drug that these are like teens in the emergency room daily. So it's not, marijuana, you don't overdose on marijuana. Heroin, you do. So my post-mortem brain collection, when I go to autopsy, I get a lot of heroin abusers. Heroin kills much more in terms of overdose. But you show up in the emergency room with a lot of problems because of marijuana. Oops, what is that? So one of the things also, the other drug I wanted to emphasize, even though I won't give you so much on the neurobiology of it today, is about heroin, because unfortunately it's now getting to epidemic proportions. And heroin overdose has increased nearly 70% in the US. A lot of kids go to heroin from marijuana, but also from prescription medications. And so we're really trying to come up with better ways to get into the schools and so on for kids in terms of really telling you the impact of even starting an opiate prescription drugs, because it's leading very quickly into heroin abuse and death. Okay, so as a neuroscientist, there are many reasons why people or teenagers obviously experiment with drugs. It's a time of your life when you're exploring many aspects. That's how we learn. That's why most of us are here. Some of our ancestors explored different things, and those that survived passed on the genes, and we're here. Exploration is not a bad thing. So experimenting with drugs, however, has consequences. But I care about what does it do to the brain. And this is just a quick biology. So the DNA gives us the blueprint in every cell, and then it synthesizes transmitters through the expression of these genes. The transmitters are released from the cell, and a receptor, and it activates a receptor on another cell that changes the activity of that cell, and they're all part of a network in terms of the neuronal pathways, and they then change behavior. And so that's the thing that we care about, is how do you go from your DNA to making these transmitters that are part of networks. And you'll hear a lot about electrophysiology in terms of cell activity, because cells communicate by transmitters, chemicals being released, and changing the activity of these cells to impact on behavior. So this is the cannabinoid receptor gene. These are the cells that expresses the cannabinoid receptor in the adult brain. The top slides we call them coronal. They're this orientation if you slice the brain in this way. This one on the bottom is a horizontal if you slice the brain like this. And you can see one thing is that the cannabinoid receptor, where the cannabis or the psychoactive components of cannabis binds, they're throughout most of the brain. In fact, it's the most abundant G protein coupled receptor we have in our brains. And it's not for smoking marijuana. So we're still trying to figure out how the cannabinoid receptor regulates signaling and transmission in the brain, but it is one of the most abundant transmitters in the brain. It's in this part of the striatum I told you about, very highly abundant throughout the cortex. This is the cerebellum, which is important for motor coordination, your hippocampus, and so on for memory. So for me, the question is not if somebody smokes marijuana, it's really what are the long-term impact of these drugs? Because this is one of the issues of mental disease and vulnerability. So for that, we use often our animal models. We also study human as well, but I'm going to focus a lot on our animal models because we can look at how adolescent drug use can impact the neurobiology when they become adults and on their behavior. I'm going to focus a little bit and give you a little neurobiology lesson on the striatum. So the striatum, as I said, is an important region for your reward, motivation, goal-directed behaviors. And the cells in the striatum are called medium spiny cells, and they project to different parts of the brain. And they are very specific in the transmitters that they express. So for example, one of these cells will express the enkephalin, like I told you, like your natural endorphin, your natural heroin, and dopamine, one of the dopamine receptors, dopamine D2 receptors, and while another pathway will express another opiate, I won't get into that so much, and a dopamine D1, but the cannabinoid receptor actually is expressed on all of them. So when we look at animals that were exposed to the psychoactive component THC as adolescents and look in adulthood, we see that there are very specific things that happen in the brain. I actually thought that we were going to see much broader effects. And even our human, we studied the brains of fetuses whose mothers smoked marijuana, and actually we find very similar things in the human brain. And we see that the gene expression of the expression of genes that are part of this cells are affected much more. And the interesting thing about those cells is that they are part of a pathway we call no-go pathway. They're important for our inhibitory control. And so the thing that is also interesting is that this reduction of the dopamine, or changes in the dopamine receptors that we see, you see in actually most drugs of abusers that take other drugs. For example, cocaine, heroin, alcohol, that this is a characteristic feature of substance abusers. So how do we look at this in human? What we did in the sense of if indeed what we see in our animals are that we have these changes in this pathway with the dopamine D2 receptor and that expresses the enkephalin genes, we've decided to bring people into the lab and young people, take their history of drug use, and look at many different psychiatric and psychological parameters, and also had them do behavioral tasks that could tease out the decisions that they made in terms of when it related to reward versus when it related to inhibitory control, and look at their genetics. And interestingly, we saw, like similar to many other to a few other groups, that genetics did play a role in people who were cannabis dependent. So for example, that mutations of their dopamine D2 receptor or their enkephalin gene was predictive of cannabis dependence. But interestingly, their behavioral trait was very distinct. And unfortunately, I didn't take this into account when I designed the experiment. And this was a major weakness because they had very strong negative affect, meaning anxiety traits were very high in these individuals in every test that we did, which of course impacts their behavior. And it impacted their behavior specifically for their inhibitory control. So we asked them to make decisions where if you see a symbol that was associated with reward, to choose that symbol. It was like a computer games. I can tell the kids about it later if anybody's interested in a specific behavioral task. While other symbols that were negative, they had to avoid them. And the cannabis-dependent people, their genetics and their behavioral trait of negative affect predicted their inhibitory control deficit. But this was actually the most surprising thing. So when you look at subjects based on just their anxiety traits, it increased their risk of being cannabis dependent just mildly, or their genetics increased at like about twofold. But it's the interaction. So we realized that is making the difference? So I should have said, actually, our control subjects also, many of them, actually most of them also smoked marijuana. They never went on to have a problem. They didn't go on to have a disorder because they did not have the genetic mutations of this particular gene, and they didn't have this negative affect behavioral trait of anxiety, neuroticism, behavioral traits. So the people who are vulnerable to cannabis dependence is a huge part of genetics, and like I said, their behavioral traits. So it's not just smoking marijuana. So that's one aspect of the striatum. So the striatum is regulated by your cortex. So your cognition is mediated through the cortex, and the prefrontal cortex sends projections there. So we wanted to look into the prefrontal cortex about what's the impact of marijuana there. And the prefrontal cortex is really important for the developing adolescent brain, because we know that the prefrontal cortex is one of the last regions of the brain to achieve full maturity, and in fact until you're about mid-20s. So don't, some of the parents know it, that their kids are still make little decisions that they're more guided by their amygdala and accumbens in terms of emotions and not about cognition. So these are some of the cells they're called pyramidal neurons in the prefrontal cortex that project, as I said, regulate connections within the cortex and to subcortical regions. So we used our animal model, and we went in and looked at, actually, my poor student went in and looked at the actual pyramidal cells, measuring the cells to look at the structural morphology of these cells. And actually, there was no difference in the length of your pyramidal neurons, either immediately following the exposure to THC or when you became a young adult. However, what differed significantly was the complexity of your pyramidal cells in the prefrontal cortex. And the black are control animals from when they're adolescents to when they're adults. And there's not much change in this part of the pyramidal neurons, but there is significant difference in the complexity. And it's only adolescent THC exposure. We didn't continue to expose them to marijuana. It was repeated low, I think it's like about a joint or so every third day was that kind of exposure. So we didn't give them heavy amounts of marijuana. It was just periodic marijuana, but it changes the morphology of the prefrontal cortex. So we know that there are gene expression changes, transmitter changes, actual cellular morphological changes by adolescent THC that last into adulthood. But the question to us is really, does it make any difference on behavior? And behavior in terms of the question of gateway, because there is big issues about whether or not marijuana is really quote unquote a gateway drug. It's tough to dissociate that in humans where friends, families, cultures make a big difference. So again, as I said, we go to our animal model. And we let animals decide themselves, do they want a drug or not? If they press it's a classic operant chamber, there are different levers in their boxes, and if they press one lever, they will get heroin. And another will give nothing, or saline, or something like placebo. And adult animals that have adolescent exposure to THC, they self-administer heroin more, irrespective of their mothers and their friends. So it changes in neurobiology, and it changes their behavior. So a question then, I didn't show you the prenatal things, but prenatal cannabis also has this effect. So we know that adolescent exposure changes adult vulnerability in neuronal system, as I said, relevant to not only addiction disorders, but other psychiatric disorders. But I asked one of my postdocs, I wanted to know, does this carry on potentially into the next generation? And so she now hates me, but we've now been studying across generations. Does it make an impact? And so I'm going to end with that because I just want to show you what germline exposure to marijuana, if it has any impact or not. And germline meaning we expose rats just like we did in when they're teens. We let them grow up to become adults. We let them fall in love. They have kids. We have other people raise their kids because the moms might have not been good and the dads might not have been good parents if they were smoking marijuana when teens. No. And so we then look at the neurobiology and their behavior. And the thing that's interesting, these are now the adults of animals who were exposed to marijuana as teens. So it wasn't that these animals never directly received marijuana themselves. Yet still, they will self-administer more heroin than normal animals. And I'm not going to go through all the behavior because it's going to be, they also show some types of anxiety type behavior and depression-like behavior. And we see that this goes on not only into the first generation, it goes even into the great-grandkids. So we're studying some of this. We also look at how does the brain look? And we see very interesting things that it is the synaptic plasticity, meaning when you measure the activity of the cells, with electrophysiology, you guys are going to go outside. There's electrophysiology set up, and you can look at the actual cells, their activity. We see that the activity of their cells in here, the striatum, is different. We also looked at genes and receptor function, and primarily we see the strong glutamatergic difference. We had studied those genes specifically. We studied dopamine genes as well, but it was biased. What we wanted to do was to look an unbiased way across all the genes in our genome to see if there's a pattern to what happens to the fact of this cross-generational effects of marijuana. And so we look at epigenetics. So epigenetics says, marijuana exposure doesn't change your DNA. It is not a toxin in that context. But what it does is changes how the DNA is marked. So your genes are either open and can transcribe and synthesize genes, or they're closed. And so, for example, I'm just looking at, to show you one, DNA methylation. And when we look at these in the striatum of these adults, like I said, have never been exposed to marijuana themselves, just their parents when their parents were teenagers, we see significant impairments in systems related to the function and organization of the synapse. So it's about synaptic connections that we see not only during their lifetime, but that transcends even to their grandkids. So we're trying to understand now what's the mechanisms for that. We also know how to reverse some of this, and we can talk about that. So I'll just end by saying that the neurobiology of addiction involves aspects related to reward, our dopaminergic cells, and our opioid system in terms of regulating hedonic state. But the addictive process does relate to the genetics and epigenetics that seems to have a long-term impact on aspects of synaptic plasticity. So how the cells communicate with each other. And so how do we normalize those, I think, are going to be really key. And we'll hear in terms of autism later, because we see a number of the genes that you see in some of the autism spectrum disorders. So with that, I will stop and take any questions.